am Teacher Yunwei. Welcome to Cancer Unraveled, Understanding Carcinogenic Genes, Part 3. We have already introduced what is known as DNA tumor virus. Do you still remember which ones there are? For example, SV40, HPV, and also the EB virus associated with nasopharyngeal carcinoma. Next, we will continue to introduce RNA tumor viruses. What are the carcinogenic factors of RNA tumor viruses? How do the oncogenes in cells become V-oncogenes in viruses that reinfect cells? We will also introduce carcinogenic genes, which may be divided into two types of changes. One is quantitative and the other is qualitative change. That is, changes in quality and changes in amount. The RNA virus was discovered in 1911 by Peyton Rous, who found that tumors could develop in chickens. So he named it Rous sarcoma virus, abbreviated as RSV. Later, it was discovered that this virus belongs to a retrovirus, which is an RNA virus and carries a so-called VSRC carcinogenic gene. The V refers to virus. Then, in 1977, Bishop and Varmus found genes similar to this VSRC in other animals besides chickens. The original RNA tumor virus did not have a carcinogenic gene, but it acquired genes from host cells through transducing, which activated the expression of these genes via the viral promoter, thus activating the carcinogenic gene. Usually, these RNA viruses have specific animal hosts, so they do not cause tumors in humans. However, they have made significant contributions to the discovery of human carcinogenic genes. Next, I will give examples of how these RNA viruses infect host cells compared to how DNA viruses infect host cells. DNA viruses have a method called non-defective transforming virus, which itself does not carry an oncogene, but it can activate proto-oncogenes in the cell and transform the cell. To illustrate, it is like entering a cell and persuading a delinquent to quickly become a big bad guy. So this process takes a little longer. Another method we call acute transforming retrovirus, which itself carries a defective oncogene into the host cell, quickly transforming the host cell. I would describe this as directly sending a bad guy into the host cell. So students can imagine which infection evolves faster: the infection by non-defective transforming virus or acute transforming retrovirus. Correct, it is acute. Acute means rapid. It directly brings a defective oncogene to the host cell. The non-defective transforming virus has a longer incubation period because it can transform the cell by activating the proto-oncogenes within. This often occurs in leukemia, also known as blood cancer. The acute retrovirus infection quickly transforms cells because it carries an oncogene. The viral oncogene is very similar to the proto-oncogene in normal cells, except the oncogene it carries has undergone mutations. And next, I will introduce what leukemia is, what liposarcoma is, and what carcinoma is. Leukemia is a type of liposarcoma. It exists in clusters in the blood. Sarcoma originates from mesoderm, which is a type of solid tumor of connective tissue such as muscle, bone, or cartilage. Carcinoma comes from endoderm or ectoderm or glands such as breast, skin, urinary and reproductive systems, and respiratory systems. Thus, 85% of cancers are classified as carcinoma. The causes of blood cancer are still unknown, but may be related to radiation, age, etc. It can be divided into acute and chronic types, and further into acute myeloid leukemia or chronic myeloid leukemia, acute lymphoblastic leukemia or chronic lymphoblastic leukemia. Acute leukemia is usually more severe than chronic leukemia, with a higher mortality rate. Typically, the treatments involve chemotherapy. These often have side effects including nausea, vomiting, fever, hair loss, lower white blood cell counts, increased susceptibility to infections, low platelets leading to bleeding, oral ulcers, and kidney or liver dysfunction, etc. Hence, unlike other diseases, leukemia does not have distinct early and late stages. Leukemia originates in the bone marrow and spreads through the bloodstream to the entire body. Thus, it is a systemic disease from the start. When acute leukemia develops, if untreated, the condition may become very severe within a few days. And patients may die within months. With the advancement of medical technology and the development of chemotherapy drugs, leukemia is no longer an absolute death sentence. It can be cured. Generally, these patients experience a more difficult time with chemotherapy, especially those with acute myeloid leukemia. In the early stages of chemotherapy, they may experience intense nausea, vomiting, and hair loss. As cancer cells crowd the bone marrow, intensive chemotherapy is required to eliminate most cancer cells and restore normal hematopoietic functions. When hematopoietic cells in the bone marrow become cancerous, it suppresses our blood formation, leading to decreased white blood cells and red blood cells, and the presence of abnormal white blood cells in the blood. This report stresses that back pain should not be ignored. It could be a sign of blood cancer. Back pain may not just be a muscle strain issue, it could be blood cancer. One woman frequently experienced back pain, and upon blood tests, it was discovered that she had multiple myeloma, a type of blood cancer. The cancer cells caused fractures, which led to her severe back pain. Initially, she thought it was just muscle soreness and sought medical help, but after blood tests, it was revealed that her bone marrow cells had developed tumors. The woman underwent the chemotherapy mentioned earlier by the teacher. Modern lifestyles due to work involve prolonged sitting or standing, leading to back pain. Typically, one might think they are just tired or experiencing muscle soreness, but later, the pain becomes so severe that they cannot turn over. So everyone must pay attention, as back pain should not be overlooked. It could be blood cancer. In the course of blood cancer, it could lead to insufficient red blood cells, resulting in frequent dizziness and decreased stamina, and even lead to fractures. If you experience symptoms of anemia, such as paleness, it is best to seek medical evaluation quickly. Returning to the transforming virus, which carries certain oncogenes, it helps us identify which genes are carcinogenic. Students may wonder, teacher, the virus is so small, and we are made up of so many cells. How can a virus have my genes? As I mentioned earlier, some transforming viruses infect host cells and extract parts of my genes into their genomes, and then lyse my cells to form virus particles. These virus particles, as we know, have smaller viral genomes, so they are prone to mutations, just like our flu vaccines, which predict different strains each year. Retroviruses are the same. Their viruses are very small, so after certain mutations, the mutated genes could be the genes they extracted from me. Originally, my genes were fine, but taken out and mutated, they become V oncogenes. Thus, the V refers to virus, and C refers to cellular. When DNA viruses or RNA viruses infect normal cells, they activate my carcinogenic genes and inactivate tumor suppressor genes. Proto-oncogenes become oncogenes, C-oncogenes become V-oncogenes. The V-oncogenes are mutated, leading to the production of mutated oncoproteins, which might cause my cells to achieve immortality. Next, I will discuss a few examples. When these oncoproteins enter the cells, they may initiate cancer in two ways. One is that the amount increases significantly without mutations, while the other is that it has mutations and its activity increases. We refer to this as a quality change. One is quantitative, the other is qualitative change. Now let me introduce an oncogene called RAS. After RAS is activated, it leads to continuous cell proliferation. Once mutated, its activity increases, constantly signaling the cell to proliferate, escaping the crisis phase. We know that abnormal cell proliferation marks the onset of cancer. The transforming virus, because it differs from normal cells, carries mutated oncogenes. RAS is one such example. Usually, the mutations in RAS could be point mutations or gene recombination. RAS itself is a proto-oncogene, and after being carried by the virus and extracted, it accumulates mutations, for instance, in glycine and glutamic acid, the two amino acids. The mutated RAS remains in an activated state. RAS itself is a G protein. What is a G protein? It is a protein that binds with GTP and GDP. We call it G protein. RAS is classified as a small G protein weighing about 21 kilodalton. In the cell, it tells the cell that it can proliferate continuously. Next, let's refer to this diagram, and I would like the students to think about it. In this diagram, we can see that G protein is in an activated state when it binds with GTP. When it loses its activity, it binds with GDP. How does it transition from GTP to GDP, and how does it go from GDP back to binding with GTP? It relies on the help of two proteins. First, I will introduce GF. GEF stands for guanine exchange factor, abbreviated as GF. It can replace GTP on RAS, allowing it to swap GDP for GTP on RAS, activating RAS. In contrast, GAP stands for GTPase activating protein. It deactivates RAS. RAS itself has GTPase activity, but the activity is low. It needs GAP to bind with RAS to activate GTPase activity and convert RAS from GTP to GDP. The teacher illustrated an example. Students, you may wake up naturally or need an alarm clock to wake you up. The GTPase activity is like the state of waking up naturally. GAP must approach RAS, like an alarm clock must be near your ear to wake you up. Please refer to the diagram to explain which situations keep RAS activated. 1. GEF mutation has no activity. 2. GEF mutation increases its activity. 3. GAP mutation has no activity. 4. GAP cannot approach ROS. Great. Students should know the answer. 2. Increased GEF activity, because increased GEF activity helps ROS's activity exchange GDP for GTP. And 4. GAP cannot approach ROS. Like an alarm clock, if it cannot approach ROS, ROS cannot switch from GTP to GDP, thus staying in an activated state. Initially, the teacher mentioned VROS, which has some point mutations. In this case, it prevents GAP from approaching ROS. Then ROS will continuously remain in a state bound to GTP. Continuous activation of ROS means it can keep signaling for cells to proliferate. This is an example of a change in quality. Next, the teacher asks the students in which situations ROS is activated. 1. ROS binds with GTP. 2. ROS binds with GDP. 3. GAP activates. 4. GEF activates. Excellent, students got it right. ROS must bind with GTP and GEF must be activated. Next, the teacher gives another example of an increase in quantity. The genes themselves are not mutated. If the teacher infects a non-defective transforming virus, which does not carry an oncogene, it can induce a massive expression of oncogenes inside the cells through its powerful promoter. For example, retrovirus activates the expression of the CM-MYC gene through its promoter. CM-MYC, students should note, the C refers to cell, while MyC is an oncogene. This means it has no gene mutation, but its expression suddenly increases, which can also trigger cell transformation. What situations will lead to increased expression of CM-MyC? Aside from being infected by a non-defective retrovirus, when your DNA undergoes a break, gene translocation occurs, and the promoter of the immunoglobulin gene binds with CM-MyC. For instance, in B cells, chromosome 14 and chromosome 8 IgH and CM-MyC undergo gene translocation. In T cells, TCRA and CM-MyC undergo gene translocation. This can increase MIC expression as the promoter of immunoglobulin genes is stronger. Gene translocation usually occurs in blood cancers. For example, as the teacher mentioned, Burkitt's lymphoma involves translocation between chromosome 8 and 14, where the CM-MyC gene of chromosome 8 and the immunoglobulin gene of chromosome 14 are translocated, resulting in a significant expression of MyC. Philadelphia chromosome is found in patients with chronic myeloid leukemia, involving translocation between chromosome 9 and 22. It forms a BCR-ABL oncogene. What is malignant lymphoma? It typically arises when normal lymphoid tissue receives external stimuli or infections, resulting in continuous proliferation of cancer cells. Usually in your collarbone, groin, neck, armpits, etc., when lymph nodes swell over 1 centimeter, feeling hard and painless, and not easily movable when touched. If there are swollen lymph nodes in many areas, the chance of malignancy increases significantly. The causes of malignant lymphoma relate to age and gender. With males being more common than females and the incidence increases with age, immunocompromised status or exposure to bacteria, viruses, or human T lymphotropic viruses, or EB virus, hepatitis C virus, or herpes virus type 8, Helicobacter pylori, etc., will increase the risk of lymphoma. Additionally, long-term exposure to pesticides, hair dyes, and wood preservatives will also increase the likelihood of malignant lymphoma. As mentioned earlier, the Philadelphia chromosome is the fusion of oncogenes BCR and ABL involving chromosomes 9 and 22. This oncogene induces a pathway involving RAS and MAPK, telling cells to proliferate continuously. Inside the cell, the left side shows an inactive c-ABL, which has a cap at its N-terminal that inhibits its activity. However, when gene translocation occurs, the N-terminal inhibitory site is replaced by BCR, keeping it continuously active. Scientists found this carcinogenic mechanism, and they developed a drug called Glivec. Glivec binds to the activation region of c-ABL, effectively inhibiting excessive cell proliferation. Glivec is a major breakthrough in treating chronic myeloid leukemia. Chronic myeloid leukemia occurs in 1 to 1.5 people per 100,000, accounting for 10 to 15% of adult leukemia cases. The typical age of onset is between 35 and 55 years. Typically, allogeneic bone marrow transplantation is the best treatment, but often finding a matched donor is challenging. which significantly increases the risk of transplantation. Thus, Glivec becomes the first choice of medication. Glivec is a targeted therapy. It does not eliminate cancer cells comprehensively like chemotherapy or radiation, which can also affect healthy cells. It is a targeted treatment approach. Initially, scientists understood these carcinogenic mechanisms because the Philadelphia chromosome is the fusion of two oncogenes. As mentioned, Glivec is the inhibitor of this oncogene fusion, occupying the ATP binding site on BCR-ABL's TK, preventing it from catalyzing phosphorylation, leading to the loss of its activation function, and thus inhibiting cancer cell proliferation. Today, we also covered RNA viruses. They infect host cells in two ways. The first type is non-defective transforming viruses, which do not carry oncogenes. They can transform cells by activating proto-oncogenes within cells, such as cAMIC mentioned earlier, which represents a change in quantity. The other type infects host cells acutely, quickly transforming cells, and carries an oncogene. This oncogene is referred to as V-oncogene. The viral oncogene is quite similar to the proto-oncogene in normal cells, but the viral V-oncogene has mutations. This is a change in quantity. We have already introduced RNA viruses. Earlier, we discussed DNA viruses. They have different infection methods. Have you all learned it?